Hello everyone, I hope you are all doing well. In our last class, if you remember, we started breathing and respiration of gases, and we've learned transport of oxygen, transport of carbon dioxide, how does it take place, like oxygen, how does it get transported from outside to inside during inhalation, and from inside to outside, how carbon dioxide is exhaled. This is what we've learned, and we also learned mechanism of respiration. Today in our class, we are going to learn oxygen dissociation curve. As you all know, and you are all preparing for NEET, so you all know it, how much this curve is important. So for your, for information and convenience, I have taken the same graph from our NCRT. So, and I'm going to explain from the same graph, and I want you all to listen to this very carefully. What is this oxygen dissociation curve and how does it help us? See, oxygen dissociation curve will tell you where does the binding of oxygen with hemoglobin takes place, and where does the dissociation of oxygen with hemoglobin takes place, at which areas in our respiratory system it happens. And then what else do we learn? Oxygen dissociation curve has a lot of clinical importance. Based on this, a doctor can find out whether our lungs or respiratory system is working fine or not, or if there is any problem with our breathing. That is what it helps us to know. Now let's find out what does this curve has. If you see, there are two parameters taken here. On the x-axis, there is partial pressure of oxygen. It is measured in mm of Hg. On the y-axis, we have taken percentage saturation of hemoglobin with oxygen. Before we learn the curve, let's know the terms. Partial pressure means what? అమ్మ partial pressure అంటే ఏంటి? లాస్ట్ క్లాస్లో నేను చెప్పినాను, partial pressure is the individual pressure of every gas in a mixture of gases. అంటే ఏంటి? Mixture of gases ఉన్నాయి. Let's say in this room we have mixture of gases. Every gas has its own pressure. With that pressure only, these gases will hit the wall of the room or hit the wall of the jar or wherever they are present. So individual pressure of the gas is called partial pressure. So partial pressure of O2 is different, partial pressure of CO2 is different. So here we are talking about partial pressure of O2. How partial pressure of O2 is influencing the binding and dissociation is what we are going to learn here. Now on the y-axis, what did we take? Percentage saturation. What do you mean by percentage saturation? First of all, saturation means binding of hemoglobin with oxygen. When I say 100% saturation of hemoglobin with oxygen means all the hemoglobin molecules which is present in the RBCs in our body has bound to oxygen. The total hemoglobin molecules have bound to oxygen and now they are ready to transport. That is called 100% saturation. It also means this. See, in the last class we've learned, or you know, the structure of hemoglobin, right? How many polypeptide chains are there? 4, and each will have one heme group. To each heme group, how many oxygens can bind? One oxygen molecules can bind. So if 4 oxygen molecules have bound to hemoglobin, it means 100% saturation. If only 3 are binding, what will you say? 75% of saturation. If only 2 are binding, you will say 50%. If only 1 oxygen has bound, you say 25%. That means there is chance. If 1 oxygen is only binding, there is chance for 3 molecules to bind. Now this example, I can give you another example. Hemoglobin molecule is not like the autos and rickshaws that we have in Telangana. అమ్మ మనం ఏం చేస్తాం ఆటోలో? If it is in Telangana autos, what happens? Anybody can sit. 2 can sit in front, 2, not only 2, 6 or 7 people can sit backside. How many number of people the driver will try to accommodate, but here it is not like that. 4 means 4. Only 4 oxygen molecules can bind to hemoglobin molecule. So that's the meaning of 100% saturation. So when these two parameters are taken into consideration, what kind of curve do we get? We get a sigmoid curve. This shape curve is called a sigmoid curve. We call it sigmoid curve, or we can also call it as S-shaped curve. Now, why did we get a S-shaped curve? See, if you look at the graph here, as the pressure is increasing, the percentage saturation also increases. That means what? Partial pressure of oxygen is very important. The more the partial pressure, the stronger the binding takes place. And where does the binding take place? Always remember, binding of oxygen happens at the alveoli level, breaking of oxygen or dissociation happens at the tissue level, because that is the purpose, right? Oxygen has to be delivered to the tissues. Then binding of carbon dioxide, where does it happen? It happens at the tissue level. Dissociation happens at the alveolar level. Why does dissociation happen at the alveolar level? Because carbon dioxide has to go out of the body. It shouldn't stay in our body for a longer time. That is what we have studied, and that is what we have learned in the previous class. Now there are certain conditions. How can this binding and dissociation takes place? They are all depending upon certain factors. What are those factors? See, for association, what is association? Association means binding of Hb with O2 forming HbO2. What is HbO2? Oxyhemoglobin. So for this we need partial pressure of O2 should be high, partial pressure of CO2 should be less, H plus ion concentration should be less, and temperature should be less. If you see, all these three conditions should be less in their concentration, and partial pressure of O2 should be more. Obviously in the lungs the partial pressure of oxygen will be more. Why will it be more? When we are inhaling, the oxygen enters our lungs. So our lungs are filled with oxygen. When gas is more, partial pressure will be more. When partial pressure of more is more, this will help in binding the oxygen to the hemoglobin molecule. Any change in this value will hinder 100% binding, or 100% saturation will not happen if there is any problem with these. Okay. Then we come to dissociation. Let's see what will happen in, or how will dissociation happen, on what conditions. What are the conditions? Partial pressure of O2 should be low, partial pressure of CO2 should be high, H plus ion concentration should be high, temperature should be high. If you see, these three factors should be high for the dissociation of oxygen with the hemoglobin. Now, where does this happen? This happens in the tissue region. Where is this happening? Association is happening at the lung region. Dissociation is happening at the tissue level. Why? Because tissue should get oxygen to carry out metabolic activities. So there hemoglobin gives away oxygen. It is giving away oxygen, but what is it picking up? It is picking up carbon dioxide. That's why you can see the carbon dioxide partial pressure is more. Why? Because the tissues have produced a lot of carbon dioxide. Obviously, when carbon dioxide levels are more, partial pressure will be more. Always remember, for binding and breaking, partial pressure is important. For binding of oxygen, partial pressure of O2 is more. For binding of carbon dioxide, partial pressure of carbon dioxide should be more. So what do we learn from this graph? We learn from this graph that binding takes place at the lungs level and breaking takes place at the tissue level. Now let's see, suppose, now this is under normal physiological conditions, this is what we get. Let's say we are not getting graph like that, and the graph is shifting towards right. That means you can see the pressure has reduced. The pressure has reduced, and let's say 80. Pressure has reduced, and so the binding has reduced. That means what? When the curve shift towards right side, binding does not takes place, but breaking takes place. Which means what? Hemoglobin loses the affinity to bind, but instead it breaks away the oxygen. That means when the curve is shifting towards right side, dissociation takes place. Hemoglobin has lesser affinity to bind, and it quickly gives away oxygen. What happens when this curve is shifting towards left? When it is shifting towards left, that means association is taking place. When the curve is shifting towards left, association or binding of hemoglobin with oxygen takes place. That means what? When the curve values or when the graph values are shifting towards left side, association, towards right side, dissociation. This is under normal condition. But under any abnormal condition, suppose the curve is shifting towards right side, that means what? Hemoglobin is not binding 100%, with oxygen. That also we can take it as a clinical conclusion. That is how doctors will deduce whether your lungs are working properly or not, depending upon these oxygen dissociation curve. So this is all about oxygen dissociation curve, everyone. Let's learn about another important value here, which is called P50. Now, what is this P50 value? P50 value of PO2 means 50% of hemoglobin has bound to oxygen. Now, they have found out, scientists have found out that when the P50 value is, or when the partial pressure value is P50 with regards to oxygen, it means 50 percentage of hemoglobin has bound to oxygen. And how much is the partial pressure? They have found out partial pressure is around 26 to 28 mm of Hg. Under normal conditions, 1 minute. Under normal conditions, 50% of our hemoglobin binds to oxygen at how much pressure? 26 to 28 mm of Hg. This is normal. Suppose in any person, the P50 value, 1 minute. Okay. Right. The P50 value is not 26, but the range is going above 26. That means what? That means the hemoglobin is having lesser affinity to bind to oxygen. If the P50 value is increasing, that means hemoglobin is having lesser affinity to bind to oxygen. If the P50 value is low, right, that means it is the hemoglobin is having more affinity to bind to oxygen. So that is P50 value. Okay, and then let's learn about Bohr's effect. There is another effect which is called as Bohr's effect. What is Bohr's effect? Concentration of carbon dioxide and partial pressure of carbon dioxide is very important for binding, as you all know. What happens if carbon dioxide concentration increases in our body, or partial pressure of carbon dioxide increases in our body, and let's say the pH is decreasing? In these conditions, 100% binding of hemoglobin with oxygen does not takes place. This is called as Bohr's effect. Okay, obviously, for binding of oxygen with hemoglobin, partial pressure of oxygen should be more. Suppose if concentration of carbon dioxide is more in the body, this will not allow the hemoglobin to bind to oxygen. That means 100% binding does not takes place. This is called as Bohr's effect, right? Everyone, I hope you have all understood about this. Let me rewind or repeat one more time. P50 value, what do you mean by P50 value? It means Hp is 50% saturated with oxygen. Higher levels of P50, which means lower affinity of Hb to bind to oxygen. Now let's learn about the next topic. And what is that next topic? Next topic is regulation. Now you all know it that any system in our body is regulated by our neural system. So which part of the neural system is controlling our respiratory system? Let's see. Okay. I have taken the same NCERT page because from here only we expect most of the questions. So let's see. Human beings have a significant ability to maintain and moderate respiratory rhythm to suit the demands of the body tissue. This is done by our neural system. Who is doing it? Who is modulating or who is regulating? Regulating means what? Regulating means managing the respiration. See, sometimes we breathe faster, sometimes we breathe slower or normally. Who is managing this regulation? We have a center, a specific center in the brain which is doing these activities. Let's see who is doing it. A specialized center present in the medulla. Now you know that medulla oblongata is a part of a hindbrain. Now what is medulla doing? Medulla is the region of the brain which is called respiratory rhythm center. Please remember this, they have asked this question in NEET. Medulla region, there is a region in medulla which is controlling our respiration. And what is it called? Respiration rhythm center. What is this center doing? It is primarily responsible for the regulation, means it is managing our breathing rate, faster or slower. Usually it sees that the respiratory rhythm center sees that we always breathe normal. By the way, let me tell you, how many times do we breathe per minute? Remember, we breathe 12 to 16 times per minute. This is what this medulla does. It sees that we always breathe in this normal range, right? And then another center is present. There's another center which is present in pons varoli. Now you know what is pons varoli. It is also a part of hindbrain. What is it doing? It is called the pneumothoracic center. What is this pneumothoracic center? It can moderate the functions of respiratory rhythm center. What is it doing? Actually, in a way, it is controlling the respiratory rhythm center. In the way, you can see here, neural signals from this center can reduce the duration of inspiration and thereby alter respiration rate. What is pneumothoracic center doing? It is reducing the duration of inspiration. See, usually we are breathing 12 to 16 times. Sometimes we want oxygen more when we are in a stressful situation or we are exercising. What happens when we exercise? We keep on panting. At that time, we need more of energy. I'm sorry, we need more of air, and we start breathing faster at a faster rate. So, who is helping us to, overcome that situation to breathe faster is pneumothoracic center. What will pneumothoracic center, send signals to the medulla saying the body needs to breathe faster to take oxygen more. So that is what it does. It keeps on regulating, especially the duration of inspiration. Apart from that, we also have a chemotaxic area. What do you mean by chemotaxic area? An area which is sensitive to the changes or the concentration of chemicals. Basically, here carbon dioxide level. If in our body the levels of carbon dioxide are fluctuating or changing, immediately this area gets activated. Remember, carbon dioxide level should not be too high, because if it is too high, then there's a problem for binding of oxygen with hemoglobin. So a chemotaxic area is situated. Where is it situated? Adjacent to rhythm center. So near medulla area, there's another area which is called chemosensitive area, which is highly sensitive to carbon dioxide and H plus ions. An increase in this substances can activate the center. As you know, these two increasing levels of these two will harm our body. So what will these centers do? They will tell the respiratory rhythm center to quickly remove carbon dioxide from the body and bring back our breathing rate to normal, right? And then apart from that, we have other receptors also. Where are these receptors present? They are present in the aortic arches and carotid arches or carotid artery. Where is this aortic arch and carotid artery? These are large blood vessels which are attached to the heart. And what do we have? There are certain special cells which recognize change in the carbon dioxide and H plus ion concentration. And what will these centers do? Immediately they will tell the rhythm center, see, there is more of carbon dioxide and there is more of H plus ions, you better take them out of the body, otherwise they will disturb our breathing rate. So then again the rhythm center will make changes. So there are four centers which are controlling our breathing mechanism, the main being medulla, and the rest of them will help the medulla, right? And another important point to remember, the role of oxygen in the regulation of respiratory rhythm is quite insignificant. That means whether the carbon dioxide levels increase, it doesn't matter. It doesn't have any, they don't control. The levels of oxygen doesn't control this regulation. If it increases, it's not a problem. Oxygen levels, if it increase in our body, it doesn't make a problem or doesn't make a difference for us. Now let's see the disorders of respiratory system. What are the disorders? There are three disorders given in NCERT and they are very important, as you all know. Let's see the first one, asthma. What is asthma? Asthma is difficulty in breathing causing wheezing due to inflammation in bronchi and bronchioles. How does a person get asthma? Because of infection, that area becomes, it swells, gets infected. Which area gets infected? If you can see, bronchi and bronchioles, there's swelling in that area, because of which a person will have trouble in breathing. And when he is breathing, there is wheezing. What do you mean by wheezing? A kind of whistle sound when that person is breathing. When he is taking in the air and exhaling the air, it gives certain kind of whistle sound. And why does a person get asthma? See, none of the researchers have not yet found out what is the actual cause for asthma. Basically, asthma is related to genetics because of, some kind of genes, as well as the environment. Some people are too sensitive. If they enter into, polluted areas or dust areas, they get quickly affected and asthma is caused in them. But actual root cause of asthma is not yet found. Let's see emphysema. What is emphysema? It's a chronic disorder. That means it keeps on becoming bad and bad. Once a person gets this disorder, it worsens if that person is not getting medicated, in which alveoli wall are damaged, due to which respiratory surface is decreased. If you remember, we've learnt about alveoli, the structure of alveoli and the blood vessels. You can see how the exchange of gases happens in the alveoli region. And we've also learned that the walls of the alveoli should be as thin as possible. The thinner it is, the easy diffusion takes place. Now in emphysema patient, what happens is this alveoli wall gets deposited with certain kind of material. Now usually this disease is seen if a person has this habit of, smoking. What happens? The material which is present in the smoke gets deposited here, and then when it deposited here, the layer becomes thick, so diffusion does not happen successfully. So you can see the respiratory surface decreases. This is one of the major causes, and the major causes is cigarette smoking, right? So this is about emphysema. And the next one is, the last one is, let me zoom it, the last one. Okay, the last one is occupational respiratory disorder. What is this occupational respiratory disorder? Sometimes we get lung infections or disorders because of the places where we work. For example, if you see in certain industries, people who are working in the industries, especially those involving in grinding and stone breaking, not only that, in chemical factories where chemicals like asbestos, aluminium, certain kind of heavy metals, in those industries, cement industries, what will happen? The dust we breathe gets accumulated in the lungs, and it leads to different kinds of respiratory disorders. We call it COPD, right? And then, so dust is produced, and defense mechanism of the body cannot fully cope with the situation. Long exposure can give rise to inflammation, leading to fibrosis. So if a person is continuously working, if that is his job, and 24 by 7 he is working in that place, what will happen? The lungs get infected and leads to fibrosis. Fibrosis is when the lungs get filled up with fibers. Not only that, all sorts of poisonous gases or poisonous substances gets filled up with the lungs, and thus causing serious lung damage. Then what is the solution? The only solution is to avoid, to stop working, or what is the other thing? Wearing protective masks, right? So if you all know it, we have only three disorders given in our NCRT. Let's learn now respiratory volumes and capacities. Respiratory volumes and capacities are very important because they have a lot of clinical importance. When we learn volumes and capacities, we will know, especially the doctors will know, how is your lung health. Is it, whether your lungs are running normal, or is there any kind of diseased condition? We can find out through respiratory volumes and capacities. Okay, so let's look at each volume and capacity. As you all know, in NCRT for NEET, it is very, very important. Definitely questions will be asked from this area. Let's see tidal volume. What is tidal volume? How do we define it? Volume of air inspired or expired during normal respiration. That means when I am teaching and when you are listening, the normal breathing that we are doing, like when we are inspiring and expiring normally, that is tidal volume. And how much is that? It is around 6,000 to 8,000 ml of air per minute. And every time we take approximately 500 ml per minute, it is 6,000 to 8,000 ml of air. Then inspiratory reserve. Here reserve means extra, something extra amount of air that we are taking. So inspiratory reserve volume means the extra amount of air that we are taking through forceful inspiration. You know, sometimes when we are doing some kind of busy activity or running along the stairs, what happens? We take some extra air. So that extra air that we are taking is expiratory reserve volume. How do we define it? Additional volume of air or extra volume of air a person can inspire by forceful inspiration. That means I'm forcing my abdominal muscle and taking some extra air like that. That is inspiratory reserve volume. And how much is that? It is around 2,500 to 3,000 ml. Expiratory reserve volume is the opposite. The amount of air that I am expiring or expelling out through forceful expiration. I'm leaving the air out. That is expiratory reserve volume. And how much is that? Around 1,000 to 1,100 ml. Right? So short forms we can call it as TV, IRV, ERV. Okay? Let's see other definitions also. The next one is residual volume. Residual here means remaining. The amount of air that is remaining in our lungs even after forceful expiration. See, I'm forcing with my abdominal muscle and taking out all the air, but still some amount of air remains in the lung. That is called the residual volume or RV. How much is that? It is around 1100 to 1200 ml. Now let's see capacities. See, capacity is how much your lung can accommodate air. Maximum amount of air that you can take in, that is your capacity. Usually we use this word, no, what is your capacity? What is your capability? Now here capacities means what is the capacity of our lungs. So that means capacity is individual. It varies from individual to individual. Now let's see inspiratory capacity. What is inspiratory capacity? Total volume of air a person can inspire after normal expiration. Means I'm expiring the air and the volume of air that I'm taking in is inspiratory capacity, which is TV plus IRV. What is expiratory capacity? Total volume of air a person can expire after normal inspiration. So normally I'm breathing in and the amount of air that is going out, that is called expiratory capacity, TV plus ERV. Then we have functional residual capacity. What is functional residual capacity? Volume of air that remains in the lungs after normal expiration. So normally I'm taking out the air out and the amount of air which is still remaining is functional residual capacity, ERV plus RV. Vital capacity. What is vital capacity? The maximum volume of air a person can breathe after forceful. See, I'm forcing all the air out and after that the maximum volume means tidal volume plus the extra amount of air that I'm taking in is vital capacity. That means extra amount of air that I'm taking in, extra amount of air that I'm expelling out, all that summed up together is vital capacity, which is ERV plus IRV plus TV. Now total lung capacity. What is total lung capacity? The total volume of air accommodated in the lungs at the end of forced inspiration. So I'm forcing and taking as much as of air, as much as I want to expand my lungs, that becomes your total lung volume, which is what we are summing up all the capacities here and volumes. RV plus ERV plus TV plus IRV is total lung capacity. You know, total lung capacity and vital capacities will actually would be helpful for an athlete or a sports person. You know, sports person, they need more of oxygen because they'll be continuously exercising or they would be participating in a certain kind of sport. They need more of capacity to fill in more of oxygen. So these capacities will actually help him improve his performances. Not only that, volumes, these respiratory volumes and capacities will help a doctor to diagnose and monitor certain lung diseases like asthma. whether his lungs are performing well or not by checking the volumes and response I'm sorry, volumes and capacities a person's lungs, whether if he is, you know, suffering with asthma, how well they are working, or if he has certain kind of lung infection, those can be monitored. Or even COPD is chronic obstructive pulmonary diseases. COPDs can also be monitored, can also be detected if we find out these volumes and capacities. Not only that, a disease can be monitored. You know, when a person is suffering with a lung disease, as he progresses, the disease will become chronic sometimes. It will become more severe. So by studying his capacities, his volumes, his lung capacities and volumes, one can find out, you know, how to reduce or how to treat that person's infection. Overall health of the lungs can be found out through volumes and capacities. So that is the applications or importance of knowing volumes and capacities. Okay, so let me, you know, as you can see on the board, this is the easy way to remember all these volumes and capacities. So you can see here, RV plus ERV is FRC. If you can write it in this way and remember, this will help you to remember things easily. RV plus ERV is FRC, IRV plus TB is IC, then ERV, IRV plus TB is VC. When we add up all, it becomes TLC. Right? So this is the end of the chapter, everyone. I want you all to go through all the videos, and in the next class, we'll also discuss PYQs related to this chapter. Thank you all.